So we earlier talked about how graded potentials will generate one after the other the longer and the more neurotransmitters bind to the chemically gated ion channels that we find all along the membrane of dendrites and cell bodies in particular. And the more graded potentials form one after the other and maybe even simultaneously, the more and more they add up in voltage. One graded potential by itself only changes the voltage of the membrane, the postsynaptic membrane, by 1 or 2 millivolts. But if we have many of these graded potentials occurring one after the other or simultaneously, they can go through summation. And as they spread, and I'll explain to you in a different video clip how it is that they spread, how the ions flow and create a current that spreads. As these graded potentials spread in the form of what we sometimes call a local current, they could potentially make it all the way to the axon hillock. If at the axon hillock these graded potentials succeed in reaching a change of about 15 millivolts, about 10 to 15, but let's agree on the number 15 millivolts. And let's say that we're going to assume that the resting membrane potential of our neuron is minus 70 millivolts, then we see that our threshold for our neuron is going to be right at minus 55 millivolts. So if summation reaches, let's say, minus 60 millivolts, we're not going to fire an action potential. If we reach an minus 56 millivolts, according to our agreement of there needing to be a change of 15 millivolts, we're still not firing an action potential. On the other hand, if our graded potentials succeed in reaching minus 55 or beyond, perhaps minus 50, yes, we will now indeed fire an action potential. In other words, this change in voltage, the moment we reach this threshold or beyond at the axon hillock, due to the summation of these graded potentials that possibly make it to the axon hillock, we then can open up the sodium voltage gated ion channels. And we're going to see then, of course, as you know by now, that sodium will rush in, because the concentration of sodium on the outside of cells is greater compared to the inside. And we see that depolarization has begun, and we have, in other words, fired an action potential. We're going to now take a look at, or in the next video clip, I should say, we will take a look at what a graded potential looks like when it spreads. We will also take a look at what one particular action potential is all about, and we will take a look at how an action potential succeeds in propagating itself all the way down an axon towards the axonal terminals. Remember, when we use the term propagation, which is typical for action potentials only, it implies that the action potential recreates itself over and over and over again. So action potentials propagate. On the other hand, graded potentials, they summate. Action potentials do not summate. Graded potentials summate. Action potentials propagate. Graded potentials do not propagate. We also say that action potentials fire because they always occur with the exact same strength, and they maintain that strength as they propagate, or in other words, as they recreate themselves, they do that with the exact same strength. Graded potentials, on the other hand are called graded because they can be stronger or weak. And we say that they generate, they do not fire.